स्लाइड शो में डालना सर। चलो ना। अच्छा अच्छा, साउंड के लिए स्पीकर, ये माइक्रोफोन। अच्छा, कैसा पता चलेगा कितना जन लॉगिन किया? यहाँ पे आ जाएगा सर, चैट में सर। अच्छा, मेरा कहाँ, मुझे कहाँ पे दिखेगा सर? आप इधर रहिए सर। कैसा पता चलेगा कितना जन लॉगिन किया? अच्छा, वहाँ पे ये है जी सर। अभी ये लिंक आपने जो भेजा मेरे पास भी वही होगा? हाँ सर। ठीक, तो मैं जस्ट इसको ऐसा कर लूँ ना? जी सर। ठीक, थैंक यू। जाते हुए दरवाजा को थोड़ा बंद कर दीजिए। जी सर। We are going to start at 11 o'clock, okay? We are going to start at 11 o'clock, okay? Okay, good morning. So we are just at 11 o'clock in the morning, and we are going to start with our today's class on hematopoietic stem cells. So thanks to all of you for coming over here for the class. And one thing possibly what you could have observed is that there is a considerable amount of lag between what I am telling you and when it is reaching to you. The stem cell biology, how it has been correlated with regenerative medicine, what are the applications of the stem cells, and then what are the ethical issues which has been associated with stem cells, because there are a lot of ethical stuff that goes with stem cells. It's not very easy to work on stem cell or doing a stem cell research, because you have to take a lot of different approvals and a lot of different clearances from different agencies, just not one but multiple of them, because there is still a lot of taboo on using stem cell technologies. So people are a little cautious about it, that they don't give a free hand to anyone who wants to do stem cell biology. And last and not the least is that the numerous applications of stem cells. Now, even in the recent time, you may have encountered that the COVID-19 infection, which is the pandemic one going on across the whole world, even now the stem cell research is going to have a lot of potential, because one of the hallmark of this COVID-19 infection is fibrotic changes in the lung and in the lung epithelium, because of the reason that there is something called the cytokine storm, and this cytokine storm is in response to the viral infections, and as a result of this, the IL-6 and interferon gamma which has been produced causes a damage of the lung tissue, and that is one of the factors that predisposes eventually to ARDS and other kind of serious complications, life-threatening complications. Now there are a few clinical studies and clinical trials which has been started in trying to inject mesenchymal stem cells, and then try to see that those cells will home into the lung tissues and going to regenerate that tissue which is kind of destroyed or kind of undergone an irreversible changes, got converted into fibrous tissue and non-functional. So what we see is that stem cell biology has a lot of potentials. Okay, and today's class, if you just kind of envision it, then it's practically about two slides. Hematopoietic stem cell is that we show the hematopoietic stem cell, and then the different lineages that actually comes from there. The erythroid lineages and the myeloid lineages and the megakaryocytes. So all of these different things. So there is one slide which has been dedicated for that, but that's a little later in the lecture today, because if we show the slide, I mean that's it, the class. So we are going to go a little slow, and now that Sudeep has already covered a substantial portion of the stem cells, so I assume that you are all aware of this one, and we are just going this class as if we are just exploring what is stem cell a little more than what is we have been learning and what we have possibly heard in the previous classes. So it's not quote unquote hematopoietic stem cell. I'm going to have a common discussion about stem cell per se, and at frequent places we possibly will be talking something which already Sudeep has discussed. So bear with me those repetitions, because a few of you may not have catch up with his classes, so it is a good chance for you to kind of revise and then go back and kind of learn it for the second time or for the third time, so that the concepts and the finer details are cemented more powerfully in your mind. So with that introductions, we are going to start with the stem cells or hematopoietic stem cells. So as I mentioned, we are going to talk a little uh, in a way that it's not just hematopoietic stem cell, but in general a little bit of more of the stem cells. Okay, so I had now shown the second slide, and most probably you're still stuck up in the first slide, so it's going to come up right now. So let it to come. So this is the second slide. This actually kind of tell you the importance of the stem cell research. So there is something which is very apparent. I mean, majority of you may have heard of it, that back in 1970s and 80s and even 1990s, there is a lot of restrictions in the stem cell research across the globe. I mean, even if you take very advanced, scientifically matured nations like United States, United Kingdom, Switzerland, Germany, I mean, everywhere there is a lot of restrictions that, working on the stem cell is something unethical. It's something like against the will of God, and all these kind of things which are not scientifically very proven. So there were a lot of problems in doing stem cell research back then, but still people could appreciate the importance of this stem cell research and its numerous applications in the biomedical sciences and so many other places, in regenerative medicines, in different scenarios where the patient needs to graft something. So there are multiple avenues where stem cell research is so fundamental that even great people. So this slide is essentially showing you that how big peoples, big politicians and policymakers and senators and peoples who kind of decide the science policy of a country are now slowly aligning themselves to the stem cell research and then appreciating its true value, and which is a very good news for us, because this has been kind of inhibited for a long time because of these different restrictions. Now it's slowly and much better right now. Right now, many places, even at AIMS, you can have a dedicated stem cell research. In fact, we have a stem cell research facility over here. Dr. Sujata Mahanti, she is in charge of that facility across the near the the CN center. So you can walk through them and then walk into that department and then actually see the different kind of logistics and the other arrangements over the stem cell facility over there. So just a few of the statements, possibly you may have read a few of them, like science has presented us with a hope called stem cell research, which may provide our scientists with the answers that been so long beyond our grasp. This is by Nancy Reagan, a great person and a big person, big personality. The second one is a very good statement that this is not a matter of using embryos. So people are skeptical that when you're doing stem cell research, possibly you're going to kill embryos, and that is unethical, against the will of God. And church and other kind of religious places has lot of restrictions, and they used to lobby not to fund science stem cell research. But then someone speaks out a volumes about that and tell that this is not a matter of using human embryo that has the potential to produce life rather than otherwise discarded embryos that has the potential to save life. Very good statement. That stem cell research, you don't have to kill an embryo, you don't have to kill a baby to do a research. Even from the discarded embryos from the IVF clinics, which either way are going to be discarded, we could salvage those embryos, isolate the cells from there, and then do a lot of fantastic good research and know a whole lot of biology, which is going to have a profound influence on medicines and clinical sciences. So it's not about live embryos. Even with the discarded and the dead embryos, you could still isolate the stem cells, and that has the capacity to save life. So that is very good statement. So this slide is essentially to tell you that what the importance of the stem cell biology, people started feeling it when slowly we started kind of venturing more into this particular subject. So this is a slide that is actually showing you, that is going to show you in the couple of seconds, the timeline of the stem cell biology development. Again, we are not going to spend so much time into that. Just going to say that it started back in 1998, 1990s, something like that, where the first stem cell was been extracted from the human embryos, and then until 2004, it's kind of $1 billion funding agencies where a lot of research activities are going on in the stem cells. And these days, I mean, almost any paper you take up in science, you will see that there are some research which has been kind of aligned with some kind of a stem cell biology. And then we know that there are something called the induced pluripotent cell, which is another little off track but kind of similar philosophy and similar hypothesis like the stem cell biology. So there are different avenues parallelly which is kind of getting influenced and uplifted because of this research in the stem cell biology. Okay, so these are all the slides which I'm trying to show you, assuming that all of you actually are already aware of this one. This is just to reinforce the value of the stem cell research and taking stem cell research as a career in your future. So this has a lot of potential. That's something which is absolutely non-challenged. Anyone is going to accept and agree with that. So now we start with what you call as the stem cell and its definition. What do you understand exactly by the stem cell? So very simplistic definition. If you go to different books on stem cell biology and other places, you are going to encounter hundreds of different definitions of stem cells. Excuse me. So the easiest way to understand a stem cell is a cell which is not committed, which is not differentiated, and which has a potential to get converted into a different cell type. So again, I'm just going to repeat it, that the cell which is not committed, the cell which is not differentiated, and the cell which has the potential to get converted into different cell type. That's what is the very easily understandable definition of the stem cells. It may not be the best definition, and you can always go back to the textbooks and look for the more definitions which is linguistically more finer and much better. But for our understanding, this is sufficient, is that we are talking about an uncommitted cell which has the potential to differentiate and form different cell types. Okay. Now from here you may have a doubt, a few of you may have a doubt, that is it that if you have a stem cell we can produce anything out of it, any damn thing? No, that's not the truth. The fact is that if you have a stem cell, we could have a few stem cells which has the capacity to produce each and every cell types in the body, and we call them with a special name which is known as the totipotent cells, and those are the very early embryonic stem cells. And majority of the stem cells that follows after these are not totipotent, but they are pluripotent, and their lineage committed potent, and they have their potency levels are different. So if you talk about a stem cell which has the maximum capacity to produce any cell type is at the embryonic stage, and after that quickly they loses the totipotent capacity, and they become more focused on producing the cells of a particular lineage. Like for instance, in today's class you have a hematopoietic stem cells which is there in the bone marrow, and you could use theoretically that stem cell to produce any blood cell type in the body, right? But what you cannot do is convert the hematopoietic stem cell into neurons, or you cannot convert the hematopoietic stem cells into hepatocytes. We cannot convert the hematopoietic stem cells into kidney cells. I mean, you could struggle it and do it artificially by using a different IPS kind of an approach, but in vivo, no. A hematopoietic stem cell is not going to get converted into a neuronal cell, so it's not going to convert it into hepatocytes. So they are more committed to a particular lineage. Okay, so that is something called pluripotent cells, because it has still the potential to differentiate into different cell types as per our definition, but it does not have the capacity to produce any cell type. Okay, and that is totipotency. So we are going to see that again in a little while. Possibly there will be a few kind of discussion on that. So as I mentioned, that these are blank cells. So the word blank is a little, I'm not very happy with that word blank, so bear with me. But that is something like, unspecialized cells, uncommitted cells, the cells which has not been in a particular differentiation lineage. That's something called the blank cells, and they're capable of dividing and renewing themselves for long periods of times, and has a capacity for proliferation and renewal. Now over here you have two very important terms over here. One is proliferation, another is renewal. So when you say proliferation and lineage commitments, I am assuming that you have been aware of this word because Sudeep has already kind of covered this class. So when you talk about lineage commitments and when you talk about proliferations, we are talking about the capacity of the stem cells to differentiate into different cell types, and therefore there is something called the stem cell differentiation process, right? And there is something also like, if the stem cell actually differentiates to all the different cell types, then very soon you are going to run out of all the stem cell pool, right? And therefore there should be a mechanism in the body to restore those precious pool of the stem cells so that we never deplete them. Like in hematopoietic stem cells, there are a few stem cells which are going to or forever remain in the niche of the bone marrow no matter what happens, because they are the one that is going to produce continuously the different lineage committed cells. So there is something called the self-renewal of the stem cells, which has been actually represented over here, and there is something called the proliferation and the lineage commitment, which is something different. And this actually brings us to a very important kind of a cell division, which the stem cells actually undergo, and those are known as asymmetric division and symmetric division. So what is that asymmetric divisions? I'm pretty sure that Sudeep may have covered it, but even then I'm just repeating it, that when you have a symmetric division means you have a stem cell that is going to renew and produce another copy of the stem cells, or is going to produce a daughter stem cell. That ensures that the stem cell pool is never been depleted out. So that is the symmetric divisions, where a stem cell is producing another stem cell. But there is scenario which is majority the case where the stem cell differentiates to form other cell types. We call as the lineage commitments, right? And therefore that's an example of an asymmetric cell divisions, where the stem cell differentiates and loses its stemness and gets committed into a lineage committed or lineage differentiated cells. Like you have the hematopoietic stem cells getting converted into say erythrocytes or say megakaryocytes, or T cells and B cells. So here when you form the T cells or the B cells, they really doesn't have the stem cell capacity, right? So we are talking about an asymmetric divisions where that capacity to self renewal has been gone, and now it has been committed to a particular step. So that is something, two different things: renewal, proliferation, and lineage commitment and differentiation. So very important, these three terms, two of them are very close related with each other, which we talk about the proliferation and the renewal, and then you have the lineage commitment, so the differentiation in the different cell types. Okay, so this is pretty much what you are talking about. This is again what I just mentioned about, that self renewal is a capacity by your cell has a capacity to continuously divide and produce more copies of itself. Now we're talking about two different terms, pluripotent and repair. What is pluripotent? Ability to produce different kinds of the cells, but again very limited. We cannot produce any cell types as a result, even if it is a pluripotent stem cell. So they are very focused on a particular lineage or particular differentiation pathway. Okay, so that is something which is very important, right? Now kinds of the stem cells. So this is something possibly we have been covered, and I'm not going to spend any time on that. So I'm going to flush through this slide very quickly and let it to come in the screen, and I'm just going to show, and then we are going to walk away from there. Okay, so we are talking about totipotent cells, we are talking about pluripotent cells, and we are talking about multipotent cells. What is the totipotent cell? It's a cell which can theoretically form any cell types in the body. The cells of this inner cell mass of the developing blastocyst has a capacity to do that, because you know that when the sperm fuses with egg, then they form the zygote, and when the zygote is formed, then it undergoes a couple of cell divisions, and then eventually it forms the morula, and from there the blastocyst, and then we have the first differentiation signal in the blastocyst, where we have the inner cell mass and you have the outer cell mass, right? The inner cell mass actually forms the embryo proper. It forms the entire cell types of this embryo. So it's kind of a totipotent cells over there. And we have the outer cell mass that is also contains stem cells, but we call them as the one that produces the placenta and extra embryonic tissues. So the inner cell mass forming the embryos, and the outer cell mass forming the placenta and the extra embryonic tissues. Each one of them have their own cell lines, stem cells, but at the same time the cells of this inner cell mass has totipotent in its nature, because they can produce any cell types of the developing fetus. But that's for a very transient point of time, and after that it starts showing into lineage commitment, and we completely lose that totipotency of that cells. So this is pretty much what has been mentioned in the slides, and you could always spend more time and read it very carefully, but we are actually just going through it because this is something very fundamental and possibly been already covered by Sudeep. Okay, so let's see over here. So this is the stem cell divisions. Again, kind of similar things which I mentioned, so we are not going to spend too much time into this, and we are now going, we kind of already discussed that one, so I just shift the slides, and then we are talking about the kinds of the stem cells. What are the different types of the stem cells? Okay, so this slide which you are right now seeing, I had already kind of removed these slides, and now we are into a different slides, which is the types or the kinds of the stem cells, where there's embryonic stem cells, we have the embryonic germ cells and we have the adult stem cells. Many people has some kind of an idea or thoughts that once you are in an embryonic stage, then only we have stem cells, and once you grow up and become adults, this adults doesn't have the stem cell pools. I mean, that's not right? Because every cell, every tissues, if you see, there are specific stem cell pools in them. Like skin, and you have the skin stem cells. We have the kidneys, and you have the kidney stem cells. We have the liver, we have the hepatocyte stem cells. So you go to every tissue, every major organ, every major tissues, and you'll see that there are a stem cell pool which regenerates those tissues and maintains them, right? So that is very important, and those are the adult stem cells. So it's not that stem cells is just a monopoly of the embryonic period. We do have stem cells also in the adult life, and those actually helps in the normal tissue maintenance and tissues renewals under different conditions, or stress, or some kind of a repair needed, some kind of a turnover is needed. So it is the stem cell pools in those tissues which actually helps to do that one. So this is what I had explained to you earlier. So we are not going to stop in the slides. We are going to talk about fertilized egg, totipotent cells, blastocyst, cells isolated from inner cell mass, which is going to form the embryo proper, and then put them into the culture dish, and then grow the cells, and then see what is the natures, whatever we wanted to study. So that's the slide which I was actually talking a little back, that how we could isolate the cells or the stem cells of this inner cell mass, which is going to form the embryo, and then find out that what are the different signals and what are the different extracellular matrix mediated cues which are being transmitted to the cells, so that they kind of go for a particular lineage commitment. So that is what this slide talks about, and this is the one which I kind of again discussed with you regarding that the different stages of the cell divisions. You could appreciate over here that it's a very symmetric cell divisions, where from the 1 cell stage to 2 cell stage, and then 2 to 4, 4 to 8, 16, 32. It's in kind of a geometric progression where you have a large number of cells which is being produced, and then you have this inner cell mass, outer cell mass scenario, where you can actually have two different. So that's the first example of a stem cell divisions, where we have two different pools of the cells with two different fates. They started with one common progenitor populations, one common ancestry populations, but then they got into two different lineages, one forming embryo, another forming the extra embryonic placenta and other kind of fetal membranes. Okay, so that is something what has been actually explained in this particular graph. So we are again moving to the next slide and see what is going on over here. So this is kind of the same thing, that now you grow them in the tissue culture flask, okay, and the whole biology of the stem cell research is actually been dependent on finding out this step over here. I don't know whether you could appreciate the mouse, but that is something over here, that where you plate the cells and then you stimulate them with the different growth factors, cytokines, hormones, small molecules, prostaglandins, lipids, sugar. There's n number of stimulators and activators and modifiers and inducers which are going to take those cells to a particular differentiation lineage, and you could understand that what are the factors which drives it, and when it happens, what are the molecular changes. So what is essentially in this slide we are talking about is that if you could harvest those cells from the inner cell mass and could grow them into culture and study their properties in great detail, that kind of gives you an idea about the differentiation program of the stem cells and what are the signals necessary for them to do a lineage commitment and go for one pathway or not other pathways, and also what are the players which actually drives this entire process. Because many of the time, one of the hallmarks of the cancer is that there is a lack of differentiation. Remember that the cancers which are very aggressive are the ones which are poorly differentiated, so they kind of retain the stemness. So somehow they get back the stemness. Now they can proliferate very fast. So if you understand that what is the mechanism to push a cells in a particular differentiation lineage, that is going to be extremely helpful for us, because we can then rescue the cell which is got trapped into a proliferation loop and continuously undergoing proliferation. We could rescue that particular cell and prevent the development of the cancer by allowing that cell to perform for a lineage differentiation pathways. And there is an example in the later on the slides where you talk about a particular type of a leukemia. Okay, and in that leukemia, how a particular agent, very small molecule, is going to cause the release of the differentiation block, which in the first place causes leukemia, and then the leukemia is cured, right? We are going to have that example very soon. So that is something which is, again, just to go back to this one slide, you could see that if you add specific factors, they could be made to differentiate into different lineages, forming different cell types or different organs. Okay, so that's the thing which we are again going to see. This one, we talked about this one in detail, and then we are actually going to go from there. Now this is the slide which kind of walks us through that the inner cell mass derived components, and how if you could plate them on a tissue culture petri dish or some kind of an aseptic conditions, then you could differentiate them in essentially all the different cell types, because these are the cells derived from the inner cell mass, and either way those cells have the capacity to produce all the different kind of embryonic cells and tissue types, which is important for the baby's survival, so that the stem cells get differentiated and form specific organs with the definite functions. And that is what is actually explained over here, that how the cells have the capacity to do this one. Okay, and we could also recapitulate pretty much of these things in vitro in a tissue culture model, where you add a particular cocktail of cytokines or growth factors or hormones, and then drive them to differentiate into a particular lineage, say formation of the cardiomyocytes, or say formation of beta cells secreting insulin, or say in the neuronal cells which has been transmitting some signal transductions, by depolarizations and polarizations. So people kind of identified all those kind of differentiation pathways. So that's very fascinating. Okay, so this is what is this slide over here. Now we are talking about another example where kind of take the stem cells and then differentiate it across neurons or neuronal cells. And one of the hallmarks of these neuronal cells is these are nestin positive cells, or in other words, if you immunostain them with nextane, then they are going to glow up, right? That shows they are expressing neuronal markers, so they are actually neuronal stem cells. And again, you could see that we could actually isolate it and then actually differentiate it along that particular lineage. So that's all this slide actually talks about. This is just an example. The previous one was another example where you could actually take and differentiate into different kind of cell types, right? Cardiocytes and muscle cells and whatever you want to, depending upon what cocktail of inducers and drivers, very important, you're going to put into it. Now over here there is an ethical issue, because why we need to have some kind of a regulation and what kind of things you are going to differentiate into. Okay, so that is something why the ethical people or the people of the ethical committee actually wants us to take a detailed review and permission from them, that what is the purpose of our research, what are you going to achieve with this one, why you are going to take the stem cells, what is the end purpose of that one, what is the expected, you know myeloid lineages. Over here you have these lymphoid lineages for producing B cells, T cells, dendritic cells, and other antigen presenting cells. We have this one with the myeloid lineages, and in the myeloid lineage again you have the two divisions. You have the granulocyte lineages, and you have this megakaryocyte and erythroid lineages, which can form erythroid cells and megakaryocytes. Both of these are anucleated cells. So you have the stem cells over here, and we have this hundreds of different kinds of these differentiated cells with the different functions. What the function RBC can do is not the function what platelets can do. It's not the function what neutrophil can do. So each one of these cells types has its own specialized function, but they start from the same pool. Now that's a very obvious question. How do you achieve it? How do you do it? What kind of changes, molecular changes are actually been happening when the stem cell differentiates into a particular lineage and does the lineage commitment? So that is something which is a very active area of research in biology. And can any one of you send a message that whether you are able to see the live thing, or it's something like which has been stuck over here? Is it audible? So hold on. Let me call these people and see that why it's not been streaming. Just give me 1 minute. I'm just going to find out why it's not streaming. अभी शायद is it streaming now? ये काफी स्लो आता है क्या? सर डिले चलता है थोड़ा नेट का भी ऐसे स्लो इशू आता है सर स्लाइड मैंने स्लाइड चेंज कर लिया अभी आया बहुत लेट चल हाँ हाँ ठीक है अभी आ गया हाँ ओके ठीक है बीच में रुक गया था so this is again the application of the stem cells in the different areas and how we are actually going to हाँ जाने का टाइम कर दूंगा how you are going to influence the way it is going to enrich this particular biomedical sciences. So this is the different applications in tissue repair, in heart disease, in cardiovascular regenerations, in cancer, Parkinson's disease, diabetes. So this is something again possibly most of you have heard of it and been possibly covered in the yesterday's class. So tissue repair, again I'm going to go and quickly slide this one, heart disease and myocardial infections and so forth. Okay, so this is the slide. Possibly I'm going to spend some time into it, let it to kind of reach to you. So okay, so this is a study where people kind of show the capacity of the stem cells to do this one, and over here if you could see that what they are actually doing is that they are injecting into the mouse heart the stem cells with the hypothesis is that this stem cell is going to regenerate this heart. Suppose there's a myocardial infections and the part of the heart tissue has been damaged irreversibly because of the reason that there is lack of oxygen and then there's a tissue injury and tissue death. Now with the hope of having those stem cells to regenerate, those means those stem cells to convert themselves into cardiomyocytes, and that's what is exactly has been been done over here by injecting into this. So this is something which has been kind of very well established. So from all these examples what I'm trying to tell you is that one of the most important demanding things in stem cell biology is to find out what is the factor, what is the driver which helps to convert those stem cells into a specific cell type. Or in other words, if you have a stem cell pool, how you are going to convert them into cardiomyocytes, how you are going to convert them into neuronal stem cells. So what cocktail of cytokines has to be added, what are the different factors that has to be added. This is something which kind of the most important question in this stem cell biology. Okay, so leukemia and cancer, rheumatoid arthritis, again I had kind of shifted the slides and we are again focusing on the one again, let it to come into a screen. So this is in the type 1 diabetes where the problem is the beta cells are actually been damaged either because of an immune mediated response or some kind of an autoimmune disease where the pancreatic beta cells have been destroyed, not producing insulin, and then in those cases you could do a beta cell transplant and can regenerate those particular tissues and start again functioning them, producing the insulins. So this is another application of this one. This is about the promises of the stem cell research and what are the different types of applications which we could have. We are not kind of we had discussed about this one in couple of slides. So we are actually heading this one fast, and this is the one which I was talking a little back. uh, Important for us for this class is, related to the hematopoietic stem cell is, you could see over here there is something called the cell division pathways, and forming from there the progenitor populations to the different populations, or the different committed populations. Over here this is a stem cell which is going to self renew itself. Over here, if you could see the cursor, it is going to self renew itself, right? And it's going to maintain the pool of it. And then we have this differentiation pathway. So this is over here a symmetric division taking place, where the stem cell pools are actually being auto generated or regenerated, right? That is what is the self renewal of the stem cell pools or stem cell fractions. And then we have this lineage commitment one. In this case of hematopoietic stem cells, we have these two major populations. One is known as the CMP, which is common myeloid progenitors, and we have the CLP, which is the common lymphoid progenitors. From the name itself, it's very apparent that one of these is going to produce the different lymphoid cells or cell types, like the B cells and the T cells and this NK cells and antigen presenting cells and so forth, has to do with the immune response. And then we have this common myeloid progenitors, which is going to be again divided into two different progenitor populations. One is known as the MLPs, MEPs, which is megakaryocytes, erythroid progenitors, which is going to produce megakaryocytes, the platelets and erythroids, or the RBCs, that progenitors. And we have these granulocyte macrophage progenitors, or GMPs. So we could see over here that there are different hierarchies of cell divisions, and each of these hierarchies are going to produce one stem cell to another stem cells. But just remember, as you are going to descend down from the top to the bottom, the potency or the of the stemness of those stem cells actually gets reduced and reduced. In other words, you could have CMP which can produce GMP and MEPs, but we cannot have a provision of getting this GMP from MEP, or in other words, you cannot walk back in time. So these are already been committed. Now you cannot have an megakaryocyte erythroid progenitor giving back to common myeloid progenitor. It's not going to happen. But we can have a common myeloid progenitor which can give rise to this too. So but these are still the stem cells. So these megakaryocytes, erythroid progenitors are still very much as stem cells. But you could see that the stemness of the stem cell is actually reduced, and that's something which is very important, because when you talk about stem cell, we need to study them in the context of the capacity of those stem cells to renew themselves and to form the different lineage committed pathways. So we call them as the measure of the stemness, which is how much they had the capacity to expand their repertoire so that they can differentiate into different and multiple cell types. And that actually goes down slowly as we are descending from the most stemness of the stem cells, like the totipotent cells, to the lineage committed cells. So we kind of lose the stemness as we are kind of going down, but nevertheless we can still call them as a stem cells. That's what I just wanted to kind of hammer into our brain. It's very important for us to understand that. Now this is a slide that actually tell you a very interesting aspect of the stem cell biology is that because of the capacity that the stem cells can auto-generate themselves, renew themselves, produce another copies of themselves, if that process actually goes wrong, then you could land up in something what you call as the different kind of cancers. Now, as far as today's class is concerned on the hematopoietic stem cells, we are more bothered about the blood cell cancers or leukemias. So over here you could see that this is a blood stem cell. Now these blood stem cells, I think most of you may be aware of, these are CD34 positive cells. What is CD34? These are actually the stem cell markers on the cell surface, right? So if you take a true hematopoietic stem cell, it is CD34 positive, CD negative, CD90 negative. So it is CD34 positive, CD90 negative, CD38 negative, lineage negative. So when you have these criteria, then it is actually a true hematopoietic stem cell. So CD34 positive, CD38 negative, CD90 negative, and then lineage negative, or there is no lineage markers. Now if you take that kind of a stem cells, and if it happens that that particular stem cell has this self-renewal capacity, and that is on and on and on, then you end up with the problem of what you call as a leukemia, because over there you are producing lots and lots of the stem cells which are not differentiating into different lineages, rather they have been just producing more copies of themselves. Okay, so that is something which has been not desirable. which is the auto renewal of those stem cells. So over here again, I should have mentioned you, possibly you may have been also aware of this in previous classes, is that when the stem cell actually divides and produces its own copy, which is this one over here, then the kinetics of this one is very slow. But when the stem cell goes into this particular pathways, lineage commitment, differentiation, then the kinetics of this is very fast. So we are talking about two different things: renewal of the stem cells and a slow kinetics, happens very slowly. And then we have the commitment of the stem cells to form the different differentiated cells, that actually takes place much faster than this renewal process. Okay, and we could see that the root cause of leukemia, many times the root cause of this leukemia is actually the problem of the hematopoietic stem cell, which is not differentiating and is actually been auto-regulating itself and producing more and more copies of this. Possibly I might have an example for this one. Let's see what has been going on in a few of the slides. So this is unknowns in stem cell research. Now another thing which comes very close to the stem cell biology is actually gene cloning. So even though we kind of covered that in a different class, but this two goes hand in hand, because whenever we do stem cell biology, many a times we might need a manipulations of the genes, right? So that we have the desired quality of those progenitor populations, and for that, cloning goes very handy with that. So stem cell biology has to be studied in context also with the cloning, so that we could do some kind of an engineering and some kind of an manipulations and can get a desired trait. And that is something why we kind of put this into together, that cloning as well as stem cell research. Okay, so stem cell needs to be differentiated into appropriate cell types before they can be clinically used. Recently, abnormalities in these chromosome numbers and structures are found in the human stem cells. So this is another thing why we need the stem cells to be screened very properly, because what happens is that when the stem cells are there and then actually been auto renewing, there is an every chances that they might harbor different kind of chromosomal abnormalities and aberrations, and we just don't want that, because if the stem cell is mutated, then all the progenitor populations starting from the stem cell is going to be defective. It's going to carry that mutations, and we don't want that. So it is very important that you take the stem cells which has been used for clinical purposes for some kind of a transplantation into some patient. We need to screen them and check very carefully if they are carrying any chromosomal abnormalities or aberrations, and if they are there, to immediately discard them, because if you use them as a progenitor populations, going to overpopulate the patient, and if that is defective or that is not functional or that has some problems, then all the progenitors coming from there will be problematic. So you just want to avoid that one. That is very important, that we do a very good screening of the stem cell populations. Okay, challenges of the stem cells. Stem cell development and proliferation must be controlled once placed into the patients, and possibilities of the rejection of the stem cell transplants as foreign tissue is very high. I mean, this is very obvious. One is that when you're going to transplant or put the stem cells, then you need to see that they are not going cancerous. So you need to very closely monitor their proliferations and other kind of things. At the same time, if you transplant the stem cells, say beta island transplant, then there is every chance that this particular stem cells from a different donor, okay, a genetically dissimilar donor, is going to be end up in having a lot of rejections by the body. So again, that is something which is very challenging in the stem cell research, that how you are going to manage these two particular things. So these two particular crises, kind of rejections by the whenever you are trying to put it in the patient, whether the patient is going to reject those stem cells which has been transplanted into his or her body. And then there are issues of contamination. So we have talked about genetic aberrations, that has to be screened out. That is very important. We need to see that whether they are not been rejected, which one the stem cells, when you are going to put them back into the patients. At the same time, we are also going to screen for them for the different contaminations like fungal contaminations, bacterial, viral, mycoplasmas, so that you don't end up in more problems than you are going to offer the solutions by doing a stem cell biology or stem cell therapy. So those kind of QCs are very important for any kind of stem cell research. So I'm going to see this one. This is something called the nuclear transfer. Again, this is not really quote unquote into hematopoietic stem cells. Possibly Sudeep may have covered it, that how exactly we kind of remove the nucleus and then actually reprogram it. So feel free to go through the slide. I'm going to cover mostly the portions which possibly I should cover. Again I'm going to go a little fast in this one. This is something, an ultrasound, a vaginal ultrasound, for about 5 to 6 weeks pregnant female, which actually shows you the fetal pole, and at the same time it also shows you a small blip over there. So this is at 5 megahertz radio frequency, and this is at a 7, a little higher radio frequency, where the resolution of the image is going to be much better. You could see over here very faintly, you could see something bulging out from here. That is actually what you call as the yolk sac. Now why are we even discussing about this in today's class? Because when you're talking about hematopoietic stem cells, then if you don't talk about yolk sacs, if you don't talk about the placenta, if you don't talk about the bones, we are possibly not doing too much of justification of our class, right? So over here, if you could see, we could see a little bit of blip over here. That is actually the fetal yolk sac. And why this yolk sac is important? Because that's the place where the first hematopoiesis actually takes place. This is a little more visible in this image. You could see over here a little more clearer, where you could see a small part of the things which has been protruding. Okay, so I am going to go through this one and rather put you into the histology slides, which kind of show you a very unique populations of the cells very early in the development, which has the capacity to produce blood cells as well as has the capacity to form the blood vessels, and we call them as the Hemi Angioblast. So these are the cell types over here. You could see over here, this is the first blood cells, and many of these cells has a dual capacity or dual properties of behaving both like an endothelial cells as well as a blood cells. Okay, now this is a very specialized region, and we are going to talk about what is this region is. And in this case, you could see that these are the different cells which are actually in the early progenitor cells, which call as an Hemi Angioblast. These are the one that actually going to produce the blood cells subsequently. Okay, so this histology is very important to for you to understand that the first erythropoiesis actually starts in the yolk sac, and over there we have these populations of the cells that actually shows those kind of an first blood cell formations. Okay, so this is a timeline which shows you that, you know, if you have gestational sac at the 5 weeks and yolk sac formations from the 5th to 13 weeks, with the fetal pole which has been developed something from the 6th to 13th week, and then with the heartbeats that actually come. So this is the kind of the different timelines of the stuff that actually happens over here. And over thing, if you could appreciate that very early in the event you have these yolk sac formations, which is almost after the gestational sac is actually formed. And this is the one where the early hematopoiesis actually takes place. Yolk sac, very important. We are not going to forget that one. Okay, and eventually, you know, the placenta takes over the functions and all these other places where the hematopoiesis happens, and eventually we have this adult hematopoiesis which actually happens in the bone marrow, right? So we know that. So this is an mouse slides of the mouse hematopoietic development, where you could see that at the different stages of this mouse gestational age, we have the different events happening. So right from the day time of which is about 6.5 days, you know, the first hematopoietic tissues actually starts reappearing, and eventually you could see that as the mouse grows up and as the gestation age or the time increases, you know, there are different places where this hematopoietic system gets scattered, and now there are multiple islands of hematopoiesis taking place in the multiple places. So first start with the yolk sacs, and then we have a little other places where hematopoiesis starts, the liver, and then we have another place where the hematopoiesis starts, like placenta, and then the bone marrow. So you could see it starts from one place and then actually slowly gets scattered into different places where multiple waves of hematopoiesis actually happens. So we are using the word wave. Why? I'm going to show you just in a few minutes that what exactly you understand with the waves of hematopoiesis. So this is the early one where, you know, there is a transient hematopoiesis and then it actually goes down. So it starts at roughly about gestational day 2 or 3, and then by 10.5 it actually dies down completely. And then we have, this is going on in the yolk sacs, right? And then we have these other rounds of this hematopoiesis which actually picks up after this, and that actually goes until something like, you know, couple of weeks before the birth. And then we have the third big wave of the hematopoiesis which actually happens near the birth and actually continues until some period after the birth. And eventually we have this final hematopoiesis which you call the adult hematopoiesis, which is going to stay forever. So we could see that hematopoiesis happens, number one, at different places in the body and anatomically different regions, and number two is that they happens in as the waves. So when you say waves, means at the different organs and the different tissues and in the different stages. So that's what exactly, it's going to go up and then actually going down, and then a new wave is going to start from a new place or a new tissues, and the third wave is going to stay. And in this way, there's different rounds of the waves of hematopoiesis. So that's very important. Now, in this particular thing, this is just in side-by-side comparison showing you the mouse and the human hematopoiesis over there. I'm just going to focus on a very specific anatomical region that is known as AGM, which is very important place because of the fact that this is possibly the place where the first hematopoiesis actually starts. This is known as the aorta gonad mesonephros, or AGM. This is the region which is a potent hematopoietic site within the mammalian embryo body, and to be the first place, that's very important, to be the first place where the hematopoietic stem cells appears. So when you talk about hematopoiesis, if you don't talk about in context of the hematopoietic stem cell, that's not correct, right? So this is the tissue, this is the organ AGM, which is aorta gonad mesonephros region, where the first hematopoietic stem cell appears, and that is where the hematopoiesis starts first, and then it goes to the yolk sacs, and then it goes to the fetal livers, and then the placenta, and then we have this adults where it actually goes into the bone marrows and hematopoiesis starts. It doesn't mean that other places hematopoiesis stops, but of course very less as compared to that of the bone marrow in the case of an adult tissue, but it starts from this particular region which is AGM. Okay, that is again another slide which actually show you very nicely the timelines with this one, the pre-hematopoietic stem cells, hematopoietic stem cells getting expanded and forming hundreds of copies of this one, and then actually it forms a lineage commitment. So this actually happens in the case of mouse, and this happens in the case of humans. You could see over here that when the hematopoietic stem cell is actually formed, that is actually formed something like about 40 days after conception. So that is something where the hematopoietic stem cell starts reappearing. It actually starts over here, if you see the primitive stick, and then you have these AGM regions, and followed by the yolk sacs, and then the fetal livers, and then the placenta, and then the bone marrow. That's what exactly I was talking a few minutes back, that how the different regions actually acts as the nucleation points for this hematopoiesis to take place at different point of time. Okay, so that's very important, and that is very interesting, which is to be actually remembered. Okay, so this is a slide that actually kind of tells you the different colonies or the progenitor populations. So technically, if you ask me, this is not something very important slide, because either way you are going to learn this one when you are going to study any hematopoiesis. There is not much in remembering the names, like BFU, CFU, blast forming units and erythroids, and colony forming unit erythroids. So these are the different progenitor populations, and then they actually differentiates into the erythroid lineages. So what has been shown in this particular image is that if it has to go and do a lineage commitment and form red blood cells, then what are the different stages it actually goes through? And these are the different stages it actually goes through: BFU, CFUs, and then the progenitor populations, reticulocytes, the nucleated RBCs, and eventually they undergoes anucleations and forms the red blood cells. Okay, that's what is actually been happening in this case. So it has to go through numerous steps or numerous rounds of cell divisions to form the different populations and the subpopulations. Again, this is a very busy slide that goes into so much detail about the cell surface markers. Again, this is just for the academic interest. From the exam point of view, it's not very important for you to know most of these lineages, but just remember this hematopoietic stem cell lineage. That is good enough for you to start with CD34 positive, CD negative, 90 negative, and CD38 negative, and then lineage negative. So that means that's in kind of in classical hematopoietic stem cells. And then as it walks down through the different lineages, you have different cell surface markers that actually shows up. So this slide is essentially showing you that in this case of hematopoietic stem cell differentiation, as the differentiation proceeds through the different stages, forming different progenitor populations, we are going to come up with the different cell surface markers. And that's obvious, because now they are actually differentiated to a different cell. But what is good for us is that we can use those stem cell markers and do very good biology out of it, because you can sort them out in a flow cytometry and study about the natures, or you could look them and do a very good microscopy by looking them under fluorescence microscopy or laser microscopy or confocal microscopy, and then study their properties a lot. So that's kind of very important why these cell surface markers are there, because they gives us a handle or a tool to purify those using an antibodies to these markers, and then purifying only that particular progenitor. Say for instance, I want this progenitor over here, lin minus, c-kit minus, SCA minus, CD34 minus, and then over CD34 positive, and then if I wanted to purify these particular fractions, I'm just going to use an antibody and is going to purify this one, and then I can get all the cells which are now been in this particular stage expressing this particular marker. So that's an advantage of this particular techniques. Okay. So this is the one which kind of we had started with, this one of asymmetric symmetric divisions. Again, very repetitions of what I told you, so we are not going to talk too much about it. It's just a repetition of this entire process, whatever we talked earlier, that there is an asymmetric division and there is a symmetric divisions. Okay, now what is important over here, just remember, because why this is important in the context of this cancer biology, is that we are almost heading towards the end of the class. So just another 2 minutes or 5 minutes and then we are done with this. So this over here you could see that there is something called that auto renewal of those stem cells. So this auto renewal of the stem cells, as I mentioned, is one of the shedding point for leukemias and other blood cancers, because if the cell actually auto renews itself and produces more copy of it, then slowly what it happens is that doesn't produces the differentiated cells, right? And you have a scenario what you call as a multi-lineages cell disorders, or you call as in pancytopenias, because what is happening is that most of the cells of differentiated cells have not been produced. If you remember back the slides which are showing in a little while back, that if you have the common myeloid progenitors, if that goes bad and if that doesn't differentiate, then what happens? All the cells downstream to that, all the myeloid cells downstream to that, the B cells, the T cells, the NK cells, the dendritic cells, everything is going to suffer, and you are not going to have all those cells. So it's going to produce a pancytopenic kind of conditions, so with a severe immunocompromised state, because you are not going to produce these immune cells. But if you have a little down the hierarchy, then you may escape a few of these populations, but still you may have cells coming out from the different populations. Now over here, I know there is a kind of a leukemia which is known as acute promyelocytic leukemia. I was just talking about in the little back that this acute promyelocytic leukemia is actually a very fatal disease if it has not been treated. But at the same time, if you treat it, this is going to be one of the disease with an absolutely fantastic outcome, because you could completely cure the disease. And the whole process is this particular disease is actually there is a PML RAR fusions. Okay, so over here this is the one which I was talking about, PML RAR fusions, and as a result of these fusions, what happens? This particular genetic loci actually becomes very inactive. In other words, there is a heterochromatin formations of this particular loci. Now, because of this heterochromatin formations of this in this particular loci, the differentiation does not take place. So we have always the stem cells which is actually auto renewing itself because of these particular fusions. Now you could do two kind of therapeutic interventions. Treat them with ATR, which is all trans retinoic acid. Wonderful results, because if you treat these individuals with ATRA, then immediately this particular block is actually been removed, and the cells actually starts differentiating and goes to the different lineages. So far they are not able to go it because there is a block because of this fusion. But now that if you have been using ATRA, then it's going to remove the block, and the cells are going to go now into the differentiation pathways. So this is this one which I was showing you, and this is the second slide which actually tells you the molecular detail what is actually been happening over here in this case of this leukemia. That over here there is a PML RAR fusion that actually kind of recruits all these different chromatin remodelers which are produces heterochromatinizations, or in other words, they produces a condensed chromatin, and as a result of this one, this entire locus becomes inactive. And you could see over here there's a cross, right? This cross actually shows you that the entire locus is inactive. It is inactivity is because of this recruitment of histone complexes which are not going to activate the gene. It's going to, because of this reason, there is an heterochromatin formations. So there's an HDAC complex recruited over there, that is SMRT, the COR, and this entire region is now been not transcribed. Now if you are adding now all trans retinoic acid or ATRA, then it kind of rescues from this situation. And once it rescues from the situation, then this differentiation block is actually been removed. So you just have a small molecules ATRA, and this ATRA is going to remove the differentiation block because of this reason of there is an PML RAR fusions. And once the differentiation block is actually been removed, then all these different activators can now come in, like HAT, which is histone acetyltransferase, COA, HDAC, then DNMTs, and therefore now the locus becomes reactivated. So that's the whole philosophy of this disease, is that in the absence of this ATRA, this region has been rendered inactive because of this mechanism we have just explained, there is a formation of the chromatin, and all you need to treat the disease is treat with ARTRA, and then that differentiation block has been removed, and now the cells can either join and produces the different differentiated cells, or they are going to get rescued from this particular pool or this particular situations where there is an auto renewal continuously going on. Now remember, these cells are also very resistant for chemotherapeutic drugs, so you cannot have a very good cure of these cells because they are pretty much resistant to a lot of different interventions. But once it differentiates, then we really don't need any drug because that particular crisis has been solved. And at the same time, now that the chromatin is more open up, it has not been condensed into dense chromatin, so now you could do a lot of interventions and you could have very good results, because the drugs or the other small molecules is very much permeable into this entire DNA. Previously in this scenario it has not been permeable. So that is something again very crucial for us to know. And with this, actually I kind of finished the today's class. So before we kind of disperse, I am going to have a very quick 2 minutes, you know, summary of this class, so before, so that we kind of follow what we are actually talking about. So the whole summary of this class is that there is something called the hematopoietic stem cell, which is a kind of a stem cell, okay, which has the limited capacity to regenerate the different cell types, but at the same time it does not have the capacity to produce any cell types, right? So it is a lineage committed progenitor population, the progenitor cells, not a totipotent cell having the entire capacity to anything. Okay, now also in the context of the stem cells, we had seen that how the stem cell has been important with multiple applications in regenerative medicines and stem cell research, in doing a lot of different fascinating things of gene manipulations and everything. So there is different avenues where there is stem cell biology as applications in cardiovascular disease, neurobiology disease, another 1 minute, huh? And all these different kind of things for, which are the potential of the stem cells. Even today, I mean, we could see that the stem cell has a lot of potential in the COVID-19 scenario, where there is a mesenchymal stem cells which are actually used to regenerate the fibrotic changes in the lungs, which is one of the hallmarks of this particular disease. Also, we had seen the different stemness of the stem cells. We had discussed about that not all the stem cells have the capacity to produce any cell types. So as we are moving down the hierarchy from the most highly potent totipotent cells down to the multipotent and then the differentiated cells, the stemness reduces. One way to check the stemness of the stem cell is going to take that cell and then actually put them into nude mice and then see whether they can regenerate all the different cell types. Say for instance, if I wanted to check that whether this particular cell with me in my hand is hematopoietic stem cell or not, what I'm going to do is that I'm going to take this particular cell and then inoculate it into a nude mice and then see that whether these mice can actually regenerate all the different blood cell types. So you have to take the mice and then actually wipe off all the marrows and then transplant this particular cell, which is believed to be a stem cell, and then see whether it can rescue that particular animal. So that is a classical gold standard way to decide the stemness of the stem cells. And then we had also discussed about the hematopoiesis and how it happens in the different waves, like it happens in the different tissue types and different cell types. It starts with the region that is known as AGM, and then we have this hematopoietic stem cells which are kind of goes into the different waves of it, and each wave actually gives rise to the next one till the adult hematopoiesis starts with. So the first we started with something called the AGM region, and then we have the yolk sac, and then we have the placentas, and then we have the fetal livers, and eventually we have this adult hematopoiesis which actually takes place into the bone marrows. And these waves are actually very important because each of these waves actually produces the cells with the different potentials, and eventually when it forms the adult stem cells, that has the maximum potential in terms of producing the pan repertoire of all the cell types which is actually being needed for maintenance the homeostasis and for maintaining the normal physiological systems. And then last but not the least, what you had seen, that the symmetric and asymmetric divisions of the stem cells, very important concept, because what happens is that if this runs out of luck, then eventually we end up in having what you call as a leukemia, where it is the stem cell which actually produces or keeps on producing lots and lots of copies of itself without going into differentiations. And you have taken one example of a leukemia, and you have seen that all you need is a small intervention by ATR, which is all transretinoic acids, and you could lift this particular differentiation block and eventually make the cells to differentiate and come out of the vicious cycles of continuously cell divisions and then put them into a differentiation pathways. So with that, we are kind of wrapping up today's class. I am pretty sure that most of you have a lot of doubts even after that. So feel free to email me, WhatsApp me, call me, whatever the way convenient for you, and then we can actually discuss those specific things. But whatever we had studied and whatever the slides is going to be, because I'm going to share with you the slides, that should be enough for you at least to answer a couple of questions which you frequently ask in the exams. Okay, so have a nice day, stay safe, and again thanks for coming this class. So bye and take care. स्लैक को बंद करके मुझे जंप ड्राइव